All right. All right, please confirm me. Am I audible to everyone? Everyone, please confirm me. Am I audible to everyone? Good morning. Good morning. Everyone, please confirm me. Am I audible to everyone? Quickly confirm. I hope I am audible to everyone very clearly. All right. All right. Okay. So now I hope every one of you very quickly recall, in the yesterday class we have completed the, transcription in case of a prokaryotes, right? Now we are starting the most important, more complex process, that is the transcription in case of a eukaryotic organism. Again, the people who are listening very first time, I am requesting you to read multiple times, then only you people can remember. At the same time, I want you to compare. When you keep comparing, it is extremely easy to understand the eukaryotic organisms. All right, now if you look into the question paper which happened last month, they have asked only one question in the afternoon shift from the transcription in eukaryotes. That too, they have asked specifically from the antibiotics. Okay, antibiotics that are blocking the transcription in the eukaryotes, they have raised the question. Anyway, that's not the problem, but we have to read this particular topic very elaboratively, and very confusing it is. Very complex it is, I can say, rather than confusing. We can say very complex. All right, let's understand. First, let me tell you the simple differences to all of you. In case of a prokaryotic transcription, how many RNA polymerases are involved, friends? We studied very clearly. There is only one RNA polymerase is involved. I believe you all people remember, in case of a prokaryotes, where the process of the transcription generally takes place, the process of transcription generally takes place in the cytoplasm. We all know that. But if you look into the eukaryotic organisms, friends, there are total 3 RNA polymerases are involved, and of course, there is no doubt, entire process of the transcription is strictly taking place where, in the nucleus only. Yeah, we'll plan that. We'll plan that. Okay. Now, all the students, please try to understand my statements very correctly without having any kind of a confusion. All of you, please try to understand. Eukaryotic RNA polymerases, including 3 nuclear RNA polymerases. It is super important that I want you people to remember it compulsory. RNA, see which RNA polymerase is involved in synthesis of what kind of RNA. Most importantly, for the matching point of view, they will ask the question which organelle it is happening. So important to remember. RNA polymerase 1, it is transcribing rRNA genes. Now question is very clear, friends. rRNA genes are actually synthesizing where? That is important for the exam. rRNA genes are synthesizing in the nucleolus. So that you have to remember for the exam compulsory. And if you look into the RNA polymerase 2, which we are going to discuss very elaboratively now, RNA polymerase 2 transcribe mRNA genes, where the mRNA is actually getting synthesized, friends. mRNA getting synthesized specifically in the nucleoplasm, matrix-like structure. And more importantly, the moment you heard the name of a RNA polymerase 3, every student remembered. RNA polymerase 3 is involved in a synthesis of all the small RNAs. Let me repeat my statement. RNA polymerase 3 is involved in the synthesis of all the small RNAs. What are those? tRNA, 5sRNA, and all other type of the small RNAs can be synthesized by the Pol3. And where? That is also in the nucleoplasm only. So friends, what's the point here? What is the conclusion here? Only rRNA is getting synthesized in the nucleolus, remaining what we can say mRNA, remaining all the type of RNAs are strictly synthesized in the nucleoplasm. There is no point of getting confused over here. And another very important statement all the students has to remember here, if you look into the prokaryotic RNA polymerase, how many subunits are there? Just 6 subunits are there in the prokaryotic RNA polymerase. Yesterday class we discussed 2 units of alpha, 1 unit of beta, beta prime, omega and sigma. I hope you all remember. But friends, when you look into the eukaryotic RNA polymerases, it contain 10 to 17 subunits. Okay, 10 to 17 subunits the eukaryotic RNA polymerase contain. You have to remember this. Now, I am going to tell you one very important statement. Okay, if you understand this particular statement, most of the concept will be very much clear to all of you. Again and again, I am telling you, if you are watching very first time this one, I am requesting you to go through the recorded again. Listen, this is the important statement I am telling you. Let's consider, friends, this is the RNA polymerase enzyme. It contains N-terminal domain as well as it contains the C-terminal domain also. Every student listen. The C-terminal domain of the RNA polymerase enzyme, it is having 7 amino acids, which we generally call it as heptad sequences. What do we call it as? Heptad sequences, which means, friends, there is a presence of 7 amino acids are present here. 7 amino acids. Now listen, why I am highlighting this point. How RNA polymerase will be active in the initiation, how it will behave in the elongation, how it will behave in the termination. Everything is depends on a mechanism called as phosphorylation. This is a very unique statement, requesting everyone to remember. What is this? How can I explain you in a very elaborative way? During the time of a initiation, the C-terminal domain 7 amino acid, listen friends, undergo minimal phosphorylation. They undergo what? Minimal phosphorylation. No point of getting confused over there. Now friends, during the time of elongation, what is happening? During the time of elongation, it is undergoing maximum phosphorylation. This is a very unique statement. They may rise in a very tricky manner, diagrammatic based question also, graphical based question also. So try to understand the concept very correctly. But during the process of the termination, the C-terminal domain of the RNA polymerase, friends, undergoing dephosphorylation. No point of getting confused, friends. During initiation, minimal phosphorylation, elongation, maximum phosphorylation, termination, dephosphorylation. Okay, you will understand automatically, but remember it. Please listen to me very patiently without interruption for the period of 30 minutes. Then only you will get an idea. Okay, it is all confusing concept. Now, what I told to all of you, in case of a prokaryotic organism, how many polymerases are there? Only 1 polymerases are there. In case of eukaryotic organism, as I said to all of you, there is a presence of the 3 RNA polymerases are present. Now, I want to tell you one important statement here. The eukaryotic RNA polymerase 1, it is also called as RPA1, and RNA polymerase 2 is called as RBP1. RNA polymerase 3 called as RPC1. Now I want to tell you common statement. What is the common statement? Whatever the core subunits that are present in the prokaryotic RNA polymerase. What are the core subunits? 2 alpha units, beta, beta prime, omega. These are the core subunits, right? Whatever the core subunits that are present in the prokaryotic RNA polymerase, similar core subunits even present in the eukaryotic RNA polymerase also. Then what is the difference? The difference is only names. I believe every student is getting my point very correctly. The difference is only names, but the enzymes will share the common properties. Again, they will ask a very simple question where most of the students are unable to answer in the exam. Which of the following organism in which the three RNA polymerases are discovered in the eukaryotic organism? I want every student compulsory remember Saccharomyces cerevisiae is the organism where we have identified these three eukaryotic RNA polymerases. Please do not get confused with the point. All right, look into this. Keep understanding my statements. The more depth you understand, more easily you can analyze the questions. Look into this, friends. RNA polymerase 2, the most important polymerase enzyme which is involved in the synthesis of the mRNA. What's the common point? Tell me. The common point is that RNA polymerase 2 will share the subunits with the RNA polymerase 3 also, RNA polymerase 1 also. There is no point of getting confused over there. Just now I told to all of you, RNA polymerase 2 having what domain? C-terminal domain. Correct, na? The C-terminal domain of the RNA polymerase enzyme, what I told you, I told to all of you that there is a presence of a heptad sequence. What is called heptad sequence? There is a presence of the 7 amino acids are present. Again, I am repeating the statement. Out of this 7 amino acid, what is the statement I want all the students has to understand? Listen everyone. Out of this 7 amino acid, some amino acids undergo the mechanism called as phosphorylation. Now, question is very clear. What is the name of the enzyme that add the phosphate group? Very important, friends. You have to remember CTD kinases. Point is very clear. And what is the name of the enzyme that remove the phosphate group? Again, you have to remember, which is called as CTD phosphatases. No point of confusion. C-terminal kinases, C-terminal phosphatases, they are generally removing, adding the phosphate and removing the phosphate. No point of confusing at all. It is easy to understand also. Now, listen, and we have discussed the similar kind of a diagram in case of a prokaryotic RNA polymerase also. Now, let's try to understand the internal structure. All the students imagine, suppose example, this is a RNA polymerase 2 enzyme. What I have done, I just did the cross section. What I did, I just did the cross section of this RNA polymerase 2. All the students understand. Whenever I do the cross section of this particular RNA polymerase, what I can observe, you only look into that. Look into this, friends. There is a channel called as a downstream channel. What is happening? Through this particular downstream channel, what is happening, friends? Through this particular downstream channel, what will enter? Listen, all the students. Through this particular downstream channel only, double-stranded DNA is getting entered. No confusion. Very, very easy to understand also. Very clear. Now, we already studied very clearly, friends. We require only one strand of the DNA as the template. So we will use one strand as the template. Every student, please listen. And we know, right, how we are going to add the nucleotides. We are going to add the nucleotides at the 3' hydroxyl direction. So can you see that? This is the channel, or this is the pore through which nucleotides can be added. Through which nucleotides can be added towards the 3' hydroxyl group. There is no point of getting confused, friends. It is very, very easy to understand. The point is very clear. Now all the students, please try to understand what is happening here. This is the channel through which RNA is going out, which we generally call it as what? Exit channel. It's very clear. No point of confusion. Now again, there was a question asked in the exam. There was a fungal toxin. All the students, please try to understand my statements very correctly. There is a fungal toxin named as alpha-amanitin. Whenever we treated RNA polymerase 1, RNA polymerase 2, RNA polymerase 3 with this particular fungal toxin, these polymerases are showing different types of sensitivities. So friends, it is very important you have to know about this. It is must and should. If you look into the alpha amanitin, listen, polymerase 2 always shows the high sensitivity, which means polymerase 2 can be easily blocked by the alpha amanitin. No point of getting confused, friends. Then if you look into the polymerase 3 will show the intermediate sensitivity. No point of getting confused. And when you observe the polymerase 1, that's also very important, friends. Polymerase 1, it is not at all showing any kind of a sensitivity. Point is very clear. Polymerase 1 not at all showing any kind of a sensitivity. It is very, very important to understand the concept very correctly. So in the exam, they'll ask us the question, arrange the following 3 polymerases either ascending order or descending order according to their sensitivity towards the fungal toxin alpha amanitin. There is no point of getting confused over there. Okay. Again, I want you to understand the point. According to the alpha amanitin sensitivity, arrange the following polymerases either in the ascending or descending order. No point of getting confused over there. Okay. So first, before understanding the process of initiation, what we have to understand? First, we have to understand the promoters. If you understand the promoters first, later we have to understand the transcription factors. If every student understand, I will show you simple animation also. If every student understand what are the promoters, what are the transcription factors, then it is extremely easy to understand the process of the initiation, to understand the process of elongation, and to understand the process of termination. So requesting every student pay super attention towards the promoter and towards the transcription factor. No point of getting confused over there. All right, now let's try to understand. Now tell me, why I have used the word class 2 promoters? Why I used the word class 2 promoters? Because they are generally coding for the RNA polymerase 2. They are coding for RNA polymerase 2. That's the reason only we call it as the class 2 promoters. There is no point of getting confused. Once again, I am recapping to all the students. In the yesterday class, we read the promoters present in the prokaryotes. I believe all the students remember. Suppose this is the prokaryotic gene, and what is this is called as INR, initiation region. I believe all the students remember. In case of a prokaryotic organism, the promoters are only present upstream. I believe you all people remember. Correct na? But if you observe the eukaryotic organisms, friends, most of the time the promoters are present on the upstream also. The promoters are present even the downstream also. There is no point of getting confused over there. Okay, all the students, please try to understand this particular statement very correctly without confusion. Listen my statements correctly. In case of a eukaryotic organisms, friends, the class 2 promoters, listen every statement that I speak very correctly. The promoters which are present in the eukaryotic organism broadly categorized into 2 types. One is called core promoters, another one is called as the proximal promoters. Now friends, let's try to understand the differences between the core promoter, between the proximal promoter. Listen, understand everyone. Core promoters located on upstream also and downstream also. This is the first point. Then, because they'll give the opposite statements and they'll make us confused in the exam. When you look into the proximal promoters only located on the upstream. See, once again I am telling you, in the exam, in order to make us confuse, they'll give the statement like this. In case of a eukaryotic organisms, promoters are present both upstream and downstream. This statement is 100% correct because it is a generalized statement. I hope you are getting me. But if you look into the proximal promoter, if they specifically asked in the exam, proximal promoters of the eukaryotes, they are only present in the upstream. If they specifically asked in the exam, the core promoters in the eukaryotic organism, again remember, they will be present in the upstream also, as well as they will be present in the downstream also. There is no point of getting confused here, and it is so easy to understand for the exam. All right, now let's focus on it. See friends, be patient and please try to understand. I need very much patience from all of you. Okay, what are core promoters? You can see that there are many core promoters are there. Is it really necessary for the exam to remember all the promoters? It's not that much important, but I suggest and I request all of you at least focus on remembering the core promoters compulsory for the exam point of view without fail. So listen friends, this is INR. You can see the picture very clearly. This is called as INR, initiation region. So on the left hand side, which means on the upstream, we have two promoters, which is called Tata box and another one is called BRE. Is it really necessary to remember the full forms? It is mandatory. I will explain you. When I am explaining, you will get a good idea. BRE is also called as transcription factor 2B binding element. I hope you are getting my point. BRE called as transcription factor 2B recognition element, that is called BRE. Now, these two are located where friends? On the upstream. But if you look into the downstream, what you can see? You can see 3 yellow color boxes. You can see 1 blue color box. You can observe 1 orange color box. How many yellow color boxes are there? 3 are there. They are called as a downstream core element. Got it? You can see 1 blue color one, which is called as motif 10 element. And you can see 1 orange color, we can call it downstream promoter element. We can see what we can call it as what? Downstream core element, motif 10 element, and downstream promoter element. So again, I'm telling you, requesting you, remember the names and remember the sequences. Sequences, that's okay. You can't remember all the time. That's okay. You can neglect the sequences, but at least remember the names. First one, tell me what is the first one? Tata box. Friends, where the Tata box is located, tell me. From the INR, it is located -28 to -31 nucleotide. Tata box is located. Simple point. Why do we use the word Tata box? Because the sequence is a TATA sequence. Point is super clear. Next one, what is called BRE? Why do we use the word BRE actually? The reason why we use the word BRE, because here transcription factor 2B will come and attaches. So from the INR, it is located -37 to -32 nucleotide sequence. You people no need to remember the nucleotide sequences. I don't encourage you. If it's possible, remember. And where the INR is located, exactly at center, I can say that it is located -2 to +4. Simple point. Now, on the downstream, how many are there? There are 3 yellow color boxes. We call it as a downstream core element. All the students listen. How many downstream core elements are there? 3 are there. Okay, you can see, right? 1, 2, 3 downstream core elements are there. Understand friends? So where they are located? They are located between +6 to +12, +17 to +23, and +31 to +33. These are the areas where they are located. Then downstream promoter located at +28 to +32. I don't encourage you to remember the sequences. Please do not worry about it. But I encourage everyone to remember the names of the sequences. Don't confuse. Okay. Again, I am telling you, we are discussing about the class 2 promoters. Why we are discussing only class 2 promoters? Understand friends, it is all about understanding. Because we are studying about RNA polymerase enzyme. What enzyme? We are studying about RNA polymerase enzyme. RNA polymerase enzyme contain core promoter and proximal promoter. Point is very clear. The core promoter, again every student remember, the core promoter located both up and down, whereas proximal promoter located only up. And I have given you all the examples of core promoters. Now you can ask me a very good question. Sir, what is the use of this many promoters? That's a very good question. What are the uses of this many promoters? Of course, so again some students will think that all these promoters are used by only RNA polymerase enzyme. No. Along with the RNA polymerase enzyme, every student understand most of the promoters can be used by the varieties of transcription factors. I hope you got the point very correctly. Along with the RNA polymerase enzyme, most of the transcription factors uses this. There is no point of getting confused over there. So, along with the core promoter, we are also having what promoter? Proximal promoter. Easy to remember. So, suppose example, this is the INR. There are only 2 proximal promoters are located, or 2 to 3 are there. One promoter is located at -47. One promoter is located at -70. One promoter is located at -105. That's all. So these are all what we can call proximal promoters, which are located only on the upstream. There is no point of getting confused over there. Easy to understand. So whatever the promoters that are located at -47, they are called as, it contains the sequence GGG, CCC. So simple sequences. Whichever located at -70 to -90, we call it as the cat box. Why? Because sequence will be like this, CAAT. So that's why we call it as the cat box. There is no point of getting confused over there, and it is very easy to understand. If you forgot this also, no problem. No problem at all. Now friends, from here I want every single student pay super attention for the exam, okay, for the lesson, okay. It's very important slide from here. You have to understand enhancers, silencers. You have to understand what are transcription factors. Without that, no one can understand initiation, elongation and termination. Now tell me, as the name itself indicating, enhancers, which enhance or stimulate the process of transcription, no doubt about it. Silencers, as the name itself indicating, which silence the process of the transcription. There is no point of getting confused over there, and it is easy to understand. Now let's understand where we discover the enhancers and where we discover the silencers. Sometimes they will link these questions with unit 2 cell biology, and they will raise the questions. So that's the reason wherever you read enhancers, silencers, antibiotics, repressors, regulators, all these we have to read extremely depth. When I show the animation, you will get a crystal clear idea. Listen. Enhancer, as the name itself indicating, right, which is going to enhance the process of the transcription. No point of getting confused. Listen, all the students, please try to understand my statements very correctly. See friends, what is this site? Understand all of you. What is this site? This is called INR, the region where the transcription will start. But understand, every student, where is the enhancer located? This is enhancer. What is the point that I want to highlight here? The point I want to highlight to all of you is that even though enhancer is located very far, see, it is INR. From the INR, nearly 72 base pair upstream, enhancer is located. But friends, even though enhancer is located very far from the INR, enhancer having the ability or capability to stimulate the process of transcription. I hope every single student is getting my point very correctly. It is having the ability or capability to stimulate the process of the transcription. So the first enhancer, because this is the statement which we can expect in the exam. The first enhancer is located or identified in the SV40 early gene. There is a gene called as SV40 early gene. In this gene, we have identified this particular enhancers. Never get confused. Now, how silencers are going to block the process of the transcription? I'll tell you in a very elaborative way. Understand, use the brain. It is all about understanding. Till today, we have not exactly understood how the silencers are blocking the transcription. But based on the existing research or based on the current research and based on the data available, we understood that the silencer is causing the chromatin, listen every student, it is causing the chromatin to coil up into a condensed, inaccessible, therefore it will become inactive. What do you mean by that? Every student understand, every student, this is the gene. Okay, suppose example, this is the INR. Okay, suppose example, this is the promoter to which RNA polymerase will comes and attaches and it start the process of transcription. No point, no problem. And where is the silencer is present? Silencer is present here. Now you see what the silencer is doing. Silencer is causing the DNA to be condensed, inaccessible. See, like this it will make it. It will make the DNA into a completely condensed form. Now, where is the promoter is located? Promoter is located somewhere here. RNA polymerase enzyme transcription factors can't recognize the promoter. RNA polymerase enzyme transcription factor can't recognize the promoter. If they are unable to recognize the promoter, how they can start the process of transcription? They never ever start. As a result, transcription completely silenced. So that is what the statement that they are saying here. The available data indicate that they cause the chromatin to coil up into a condensed, inaccessible, and therefore it is in the inactive form. Very simple point, friends, easy to understand. The more depth you remember, more easily you can answer the question. The first enhancer is located on the SV40 early gene. Very clear cut point. And where the first silencer is located or discovered. The first silencer is discovered in the yeast chromosome 3. The first silencer is discovered in the yeast chromosome 3. In the yeast chromosome 3, we have identified 3 silencers, MAT, HML and HMR. Just remember that enough. Now, most important, transcription factors. So from now onwards, the process of initiation will start, the process of elongation will start, the process of termination will start. Actually, the process of prokaryotic transcription is completely different compared to the eukaryotic transcription. It is completely different. So pay super attention towards the concept. Okay, before starting this, again I am recalling a point. What is the point that I am recalling to all of you? The point I am recalling is, this is RNA polymerase enzyme. Consider this is the C-terminal domain. What is the point I am recalling? During initiation, minimal phosphorylation. During elongation, maximum phosphorylation. During termination, dephosphorylation. These 3 points I want you to remember. Now I am going to explain you. Okay. Every student very quickly answer my question. Every student very quickly answer. What is the name of the protein that guided the prokaryotic RNA polymerase enzyme to recognize the promoter? Tell me, Manju, what is the name of the, what we can say, protein that guided the prokaryotic RNA polymerase enzyme to recognize the promoter? What is that? Every student. Everyone. What is the name of the protein that that help the RNA polymerase to recognize the promoter in prokaryotes? Of course, it is a sigma factor. Correct, na? But friends, in case of eukaryotic organism, is there something called a sigma factor? No. No. In case of a eukaryotic organism, there is a no sigma factor at all. So now what you have to remember, you have to understand that the process of initiation, the process of elongation, the process of termination, everything is exclusively depends on transcription factors. Let me repeat my statement. In case of a eukaryotic organism, the process of initiation, the process of elongation, the process of termination strictly depends on only the transcription factors. Never ever get confused with the statement, friends. Now, there are two type of transcription factors are there. It seems to be a little bit complex, but when I show you the animation and if you revise 2 to 3 times, it becomes very easy to all of you. There are two type of a transcription factors are there. One is called as general transcription factors. Another one is called promoter specific transcription factor. No point of getting confused over there. What's the difference? As the name itself indicating, general transcription factor, because they are not much efficient. So I am not at all saying that by general transcription factors the process of transcription never takes place. The process of transcription will takes place, there is no doubt about it, but the process of transcription will occur only at a basal level. A minimal level of the transcription will occur with the help of general transcription factors. Point is clear. Then what is called as promoter specific transcription factor? They are more specific. They are required for maximum level of the transcription. I hope the point is very clear. They are required for what? A maximum level of the transcription. No point of getting confused over there, and it is very easy to understand. Very easy it is. Now friends, I'll explain to all of you in a different way. So you remember how to understand. You remember how to understand. Listen to me first. What are the transcription factors involved in the initiation? First, if you understand that, then we will read the function, then I will show you the diagram, then it is super clear to all of you. I'll explain twice, no problem at all. Slowly understand the concept. It's a little complicated process, very complicated, I can say that. So then now, in case of initiation, transcription factor 2. Why we use the word transcription factor 2? Because all are coding for the RNA polymerase 2. That's why we can call transcription factor 2. So the number 1 transcription factor is transcription factor 2D. Number 2 is called as transcription factor 2A. Number 3 is called as transcription factor 2B. Okay. Number 4 is called as transcription factor 2E. Number 5 is called as transcription factor 2H. I'm sorry, transcription factor 2F. Then the number 6 is called as transcription factor 2H. So total how many transcription factors are involved, friends? Total 6 transcription factors are there. 2 D A B E F H. You make your own mnemonics. You people can remember according to your convenience. But I'll tell you how easily you can remember. But you have to understand it is possible with practice. Now first I'll explain one by one. I'll show you the diagram. Please be patient and pay attention towards the concept. Don't deviate from the concept. Okay. Now the transcription factor 2D. Okay. The transcription factor 2D. The transcription factor 2D it is made up of two proteins. One is called TBP protein. TBP protein means a Tata binding protein. Then another one is called TAF, transcription activated factor. So let me explain you in more simple way to all of you. So friends, this is the gene, this is INR example, okay, and this is called as Tata box. Simple point. Now friends, transcription factor 2D made up of how many proteins? Two proteins. TBP protein, TAF protein. TBP protein will come and attaches to the Tata box. So simple. And what about the TAF? TAF will come and attaches to the INR. That's all. So simple to understand. That is called transcription factor 2D. Now next one, what is called transcription factor 2A? I will tell you, it is a kind of a linker transcription factor it is. What is the meaning of linker transcription factor? All the students has to understand. Linker transcription factor means, listen all the students, it is stabilizing between transcription factor 2B and TBP. No point of confusing friends. Easy to understand. I'll explain another time. Transcription factor 2A called as linker transcription factor. It is linking between transcription factor 2B and TBP. But we have not studied transcription factor 2B. Now what the transcription factor 2B will do? Anyway, all the students please look into the picture. Easy to understand friends. Can you see that? This is the INR. This is what? A Tata box. To the INR who will come and attach? Sorry, to the Tata box who will come and attach us? TBP. To the INR who will come and attach us? TAP. Transcription activated factor 2. Together what do we call it as? Transcription factor 2D. No point of getting confused over there. Now, what is the next third factor friends? All the students focus. Transcription factor 2B. I can tell you friends, remember like this. SS, which means transcription factor 2B involved in the start site selection. In other words, where the INR is located, in simple words, where the INR is located, that can be checked by this particular transcription factor 2B. There is no point of getting confused over there, and it is very easy to understand. Next one, transcription factor 2F. I want all the student has to understand this point very correctly. Transcription factor always associated with RNA polymerase 2. Never ever get confused with the statement friends. Transcription factor 2F always, every single time, it is associated with what? RNA polymerase 2. Never ever get confused with that particular statement friends. Always connected with the RNA polymerase 2. Okay, no point of getting confused. In other words, I can tell to all of you that transcription factor 2F having a sigma-like activity. What activity is having? A sigma-like activity. So they can ask the question in the exam, which of the following transcription factor having this sigma-like activity, which is the transcription factor 2F. Then next transcription factor called as transcription factor 2E. I'll show you the diagram, don't worry. Now, what is the ultimate function of the transcription factor 2E? It is only responsible for recruiting transcription factor 2H. Transcription factor 2H. Among all the transcription factors, the most important transcription factor is the transcription factor 2H. Why transcription factor 2H is very important? Listen why it is. Because it is having a helicase activity, very versatile, very unique type it is. It is having a kinase activity, it is having ATPase activity, and it is also having the most important and unique statement. It is involved in a repair mechanism called as a nucleotide excision repair mechanism. It is involved in where? Nucleotide excision repair mechanism. No point of confusion at all. That's the reason transcription factor 2H is said to be the most important. Now let me summarize, friends. I'll summarize to all of you. Okay, I'll summarize with the help of diagram. One second, it will load. Please try to understand, and I know most of the students who are listening first time, 100% you will get confused. 100%. Okay, so read again, it will become easy to all of you. Okay, I'll explain to all the students here. Please follow me, please follow me. So first, what is the first factor? Transcription factor 2D, correct na? Just follow me correctly. Transcription factor 2D. Transcription factor 2D contain what? TBP and TAF. You only look into this, friends. TBP will come and attaches to the data box. TAF attaches to the start point. Crystal clear, no confusion at all. Then which is coming, friends? Then you can see that transcription factor 2A is coming. What is the function of the transcription factor 2A? I told you it is linker. It is making the interaction between the transcription factor 2B and TBP to become stable. So by that they both attaches very tightly with each other. No point of confusion at all. Then the transcription factor 2B, what is the function? Can you see that? Start site selection. It is involved in the start site selection. All the students pay attention, understand that. Now finally, which enzyme is coming? Versatile enzyme, the most important unique enzyme, RNA polymerase 2 is coming. And what I told to all of you, whenever RNA polymerase 2 enzyme is coming, all the time which will be associated with the RNA polymerase 2? Transcription factor 2F. That's the reason I'm telling you. Transcription factor 2F act like a sigma. Means, now you can ask me very clearly, sir, what exactly transcription factor 2F will do? Listen all of you. Transcription factor 2F will help the RNA polymerase 2 to specifically bind to the promoter. For the specific binding to the promoter, we require the F. If transcription factor 2F is absent, how RNA polymerase comes to know that where exactly the promoter is located? So for the specific binding of the promoter, we require the transcription factor 2F. So easy to understand. Then who is coming into the picture? Transcription factor E comes into the picture. And friends, what the transcription factor E is doing, friends? Transcription factor E is involved in the recognition of H, or in the recruitment of H. Now every student, please understand my statements. This is the most unique slide. You all require a wonderful understanding. Look into that, look into that. The more nicely you understand, more confidence we get in while solving the question. Friends, look into this. You tell me, if I want to start the transcription, you tell me what should I do first? First I have to open the double-stranded DNA, correct or not? If I want to open the double-stranded DNA, what enzyme I required? Helicase. But we don't use any helicase at all. Transcription factor 2H itself act like a helicase. Itself act like a kinase. Now look into this, look into this. Now see this. What is the difference, friends? Here, what is the difference? I have written a word minimal initiation complex. I have written a word active initiation complex. Now you can ask me, what is the exact difference between the minimal initiation complex, active initiation complex? What is the difference? The difference is here. Can you see that? This is the C-terminal domain of the RNA polymerase enzyme. You only tell me, does it undergo the phosphorylation? No. It is not undergo the phosphorylation at all. And do you think double-stranded DNA is opened? No, it is not opened. That's why we can call it minimal initiation complex. Why we are calling minimal initiation complex, friends? Because transcription factor 2H not comes into the picture. It not yet come into the picture. Now look into this. Transcription factor 2H come into the picture. Look into this beautiful slide now. You can see the picture very clearly. Now you can see that there is an opening of the double-stranded DNA. There is an opening of the double-stranded DNA. Not only that, friends, you can see that. the C-terminal domain is minimally phosphorylated also. This is called as active transcription complex, which indicates that the process of initiation is completed. When you can say initiation is completed, we can say that whenever the transcription factor 2H comes into the picture, when it opens the double-stranded DNA by acting like a helicase, whenever it add the phosphate group to the C-terminal domain by acting like a kinase, then only we call it as active initiation complex. There is no point of getting confused here. Very, very easy to understand. That is initiation. Now I want all the students has to understand. Now it will become little more confused. Now we understand the initiation, very clear. Now in the initiation there are 6 initiation factors are involved. When it comes to the elongation, elongation means, you know very well, elongation means what, friends? Elongation means what? Just addition of nucleotides, nothing else. Correct na? Adding nucleotides. In the elongation there are 3 factors are involved, friends. I always encourage you people to remember the short names. Never worry about the long names. DSIF, NELF, PTEFB, then transcription factor 2S. It is little confuser concept. Use the brain and try to understand. DSIF, NELF, PTEFB. Listen, all the students, use your brain. Listen, understand. Suppose this is INR, okay? This is INR. Understand, friends, very clearly understand my concept. Now, this is RNA polymerase enzyme, okay? Now, RNA polymerase enzyme crossed the INR, starts synthesizing the RNA also. See this? RNA is coming out. It crossed the INR, see, it crossed the INR, starts synthesizing the mRNA also. Whenever RNA polymerase enzyme, after crossing the INR, there is a site is located, which we can call the pass site. What does it mean, friends? Imagine like this, you are driving a car at 100 km per hour. You are driving a car at 100 km per hour. You are coming at a very high speed. But you tell me, whenever on the national highways, whenever a toll gate come, what is the speed of your vehicle? The speed of vehicle will become 0. Because you have to stop compulsory. Correct or not? What we can call it as? Pass site. You can't move further. Same thing will happen with the RNA polymerase enzyme also. See, this and all you can't see in the animation. Please try to understand me correctly. Look into this. RNA polymerase enzyme will be passed at a specific pass site. Where this pass site is located, lying 20 to 50 base pairs downstream to the INR. Simple point. Whenever the RNA polymerase enzyme crossed the INR and reached 20 to 50 nucleotides downstream, it immediately stopped over there. Immediately. Why? Because there is a pass site. Now you have to understand. What happens in the toll gate? Whenever your vehicle stopped, what happened? You will pay the toll fees, then only your vehicle will move. Then what happens to the RNA polymerase? Look into this beautiful diagram now. If I want to allow the RNA polymerase 2 enzyme wants to move, what has to happen? The phosphorylation has to happen. Look into this. Here what kind of a phosphorylation occur? Minimal phosphorylation occur. Now all the students focus on the diagram. This is the INR, correct or not? This is the INR. It crossed the INR and it was stopped at one particular point. Now how can I make it move? I can make it move by the mechanism called as maximum phosphorylation. This is the unique point. Now you see that when the RNA polymerase is completely phosphorylated at their C-terminal domain, it can easily pass that passing site and it start continuing the RNA synthesis. All right, is it clear, every student? Now we'll stop now because it's a very complicated process. I'll continue in the next class. Is it clear? Requesting everyone look into the PPT once again and revise the class once again. Then only you will get a good idea. Is it clear, everyone? Anyone having any doubt so far? Please let me know. If you have any doubt, I will resolve it. Clear? Revise once again without fail. Then you'll get a clear-cut idea. You can ask any kind of a doubt. All right, revise once again. I'll see you in the next class. Okay. Thank you all. Please revise it. Please revise.